Once we begin the vote, the buttons will start flashing and will continue to flash even after you have entered your vote. So please press the button firmly that corresponds to your vote. If you're unsure of your vote and you wish to change your vote, you may press the corresponding button until the vote closes. And I believe the vote goes on for about 20 seconds or maybe 30 seconds. After everyone has completed their vote, the vote will be locked in. The vote will then be displayed on the screen, and Cicely will read the vote from the screen into the record. And then, and this is the most important part, it's not what the vote says, it's not the tally, it's not winning or losing. I'll go around the room and everybody needs to explain what influenced their vote, whether it was a no or a yes or an abstention, because more important than the vote is what your rationale was, what you were thinking, because that's what's most valuable to the FDA in making their final decision. There's one question, and again, the denouement of the entire afternoon is your explaining why you voted the way you did. And it could be that your rationale is even different than what you voted, but that'll be important for the FDA. Okay, so I'm going to read the voting question into the record. Does the available evidence provide substantial evidence of effectiveness of Diramicel for the treatment of cardiomyopathy in patients with DMD? And your choices are yes, no, or abstain.
Increased 14%. Impression growth was healthy across all regions, driven by
Yep, everybody can, the clock starting.
ad load optimizations. Global average price per ad increased 12% year over year, driven by ad performance gains, improvements in macro conditions relative to Q2 of last year, and currency tailwinds. This was partially
I don't see where the button is.
Yeah, I don't either.
I'm not sure how people online vote.
They send their email.
I think we just said, I think
you send an email in.
Send an email out, I think is what
Oh.
Okay, the people in the room have this little panel, and you have flashing lights with yes, no, and abstain.
No, I
abstention.
only wish I had flashing lights. We'll send they're supposed to send an email back.
We'll see what we can do next time.
To whom do we send the email?
Instructions. So can you check your email? Can you check your email? Their voting instructions?
Yeah.
You can send it to me.
Who's me? Is that Cicely?
Yes, that's Cicely.
Sending her an email right now. No, I vote no. Shkreli votes no.
See, I gotta get your email.
And included $2.4 billion in charges related to legal proceedings and $1.2 billion in the May 2026 headcount reduction. Year-over-year growth was primarily driven by increases in employee compensation,
What's the vote?
infrastructure costs, legal-related costs, and third-party AI token costs.
Get the
Excluding
vote.
the previously mentioned severance expense.
It's not working.
Employee compensation
It just says
technical
can
hires
select again and it's still blinking.
Yeah, then no, that's fine. If you put it in there, let me make sure. Did the votes register? Yeah, that's okay. It keeps flashing because you can change your mind until they stop flashing.
Got it.
Cicely, I just sent an email.
All the votes.
If you could confirm.
It takes a little bit longer because some people are online.
We expect the majority of impacted employees will no longer be captured in our headcount by the end of Q3 2026. Second quarter GAAP operating income was $18.8 billion, representing an increase year-over-year and a 31% operating margin. Excluding the Q2 legal charges and severance expenses, our second quarter operating income would have increased 9% year-over-year. Our tax rate for the quarter was 16%. Net income was $15.8 billion, or $6.18 per share. Capital expenditures, including principal payments on finance leases, were $31.1 billion, driven by investments in servers, data centers, and network infrastructure. Free cash flow was $784 million. We ended the quarter with $90.3 billion in cash and marketable securities and $83.7 billion in debt.
Where's the fucking button?
Now to the business performance. There are two primary factors that drive our revenue performance: our ability to deliver engaging experiences for our community and our effectiveness at monetizing that engagement over time. On the first, we continue to see significant gains from our content recommendation initiatives. On Instagram, global time spent this quarter grew.
What is happening? You guys voted or not?
This quarter, largely driven by improvements to our feed and Reels recommendations. On Facebook, video time spent increased 9% globally year over year, and over 10% within the US and Canada, where it was driven by ranking improvements.
I do want to emphasize this is not really an election, even though I know everybody's treating with bated breath as if it is an election. It's not a winner or a loser. It's getting a sense of the committee.
What?
And generating better training.
The vote down.
Second, LLM
Bitch
powered
ass.
agents are also
It's not an
helping with
election.
engineering development by evaluating content quality, detecting trends, and testing ranking changes. Earlier this year, we reached a milestone of every public
They have
Reels
buttons.
post
It's
on
a button
Instagram
thing.
being automatically processed through an LLM and analyzed across dimensions from topic to tone. And we're working towards including more surfaces on Facebook as well. These signals can then be passed to downstream applications across ranking, recommendations, and content policy enforcement, which is a key building block toward greater personalization.
You're the whisper?
This quarter, we also began using our Muse family of models to conduct content understanding across signals like video topic classification and summarization, and we've seen positive early results. Finally, our recommendations are also becoming more
there we go.
surfacing more fresh content.
3 yes, 9 no.
More direct control over what they see.
For the record, we have 3 yeses, 9 noes, 0 abstained.
The 2 patient reps voted
Okay.
yes.
Now here's the meat of this, and this is going around and discussing what influenced your vote. Why don't we start with Dr. Coffey, since I know he has to leave early.
Content to user matching.
Yeah,
We are now bringing
Chris Coffey, I voted no, yes, basically for the reasons discussed. I feel like the LVEF has too many questions with the sensitivity now and choice of data. The upper limb, as we discussed in the discussion topic, is a little more favorable, but I'm not convinced that it works. So with the
On Instagram feed, over half of all recommended content is now less than 1 day old, more than double from a year ago. We're also giving people more direct control of the content they see. Today, Instagram users can visit the Your Algo page, which lets users write natural language prompts to tune their recommendations. Similarly, on Facebook, we launched Shape Your Feed. Early results show.
I feel we didn't adequately discuss it. And also, I had major concerns about the FDA reviewers' report about its reliability. And the reason why I say that is because so much of it is devoted to identifying technicalities and procedural errors and using them as a basis for dismissing entire data sets out of hand without even examining them. Again, I'm still not clear on why we're using SAP 1.1. And I'm a little alarmed at how little attention is actually given to evaluating the efficacy of the therapy based on the data that was submitted. The data needs to be examined and evaluated. Furthermore, decisions made by the sponsor are often characterized in the document as arbitrary and baseless. But this seems to me to be an inaccurate characterization. From my perspective, the perspective of my community, the stakes are really too high for us to be sitting here quibbling over technicalities. A decision must be made, and this outcome is too important to let it be decided by default on the basis of a technicality. I don't think the community or the public was well served with this discussion because so much time was devoted to discussion of submission dates, who signed what, and a singular fixation on 1 or 2 patients. So not really a representative sample of all the committee members there. Again, this matter is just too important and the stakes are too high to write off the HOPE-3 trial data. And I've often heard, well, We want better for your community. It's not, you know what? I'm content with good enough, right? So if it looks promising, let's exercise that regulatory flexibility, because this is a rare disease with an unmet medical need, and that really ought to be taken seriously. Thank you.
Ms. Dunn?
I'm Deborah Dunn, patient advocate. I did vote yes for many reasons. Am I 100% vested in that? I don't know. One, I think I'm a little disappointed, and there's probably not much can be done about the small population that's in this trial. I've sat on much larger panels and sit on DSMB boards that really dumpster dive into what happened to each patient and everything. So the discrepancy between the FDA's data and the sponsor, all of that was in the back of my head. I do know going back to collect more data is going to possibly put years on this. I don't know if it can be fast-tracked, but that listening to the patients and to their families. I'm a mom. I have a heart. They don't have an alternative. And one of the speakers said, when her friend asked her, are you going to do the trial? Said, if you do nothing, you know the outcome. And that you asked what was a pivotal decision. I'm a mother of 3, and all 3 of my children were critically ill, at each, thank God, at different times in their lives. And my oldest was in a clinical trial for a disease that caused aneurysms, and I had to draw an envelope, and they were treated with what they thought might happen to be positive for this, and he skated by that. When my second son came down with it, it was already protocol, and it was because of a clinical trial. And so I'm very grateful for that. When I was trained by the FDA many, many moons ago, it was always drilled into our head, risk over benefit, benefit over risk. And so that's why I voted yes. Thank you.
Thank you. Dr. Tift?
Yeah, Cynthia Tift, NIH. I voted no because the question, I think, is rather narrow, and I don't think that the data is on this particular issue with the cardiomyopathy is that compelling. It pains me because I think that probably if this were a broader question that also involved the upper extremity, and I think some of the compelling testimony from the families, give me the impression that, if it was a bigger study, if it went for longer, if we chose different endpoints, that there probably is something there. But given the question that was asked, I think the evidence is really not that compelling. And I say that as a researcher who is in the middle of a gene therapy trial for an even rarer disease that is uniformly fatal. So I get it, as far as how this impacts families, because I'm in the middle of this with a slightly different disease. But I just, at this point, can't, with this narrow focus on what the question is, it's hard to vote yes.
Great. Dr. Wittes?
I voted no for much the same reason as Dr. Tiff and Dr. Coffey. I saw the word of the day seems to be fragile. The results were very fragile depending on how one imputed individual patients. And percent of these patients that's normal. But I would have liked to have seen some exploration of assumptions regarding who was missing. So I simply didn't see enough evidence of benefit to vote yes.
Dr. Teerlink?
John Teerlink, I voted no. First of all, the sponsors' own analyses say that they did not meet their endpoint, so with the p-value 0.09. So they did not, to address the cardiomyopathy issue, they did not show evidence of meeting that endpoint. So that was pretty easy. In addition, I think the biggest concern and something that people need to look at is if you're increasing left ventricular volumes, that is doing harm. And, that needs to be considered further as programs go forward.
Second, we have high confidence in
Okay,
our ability
I'm going to go
to
last.
capacity
Dr. Dodd.
to scale and build on
Yeah, Lori Dodd, I voted no for many of the reasons stated. This is also a biomarker for cardiomyopathy there was really wasn't clear. It's prognostic, but it's not been shown to have, I didn't see any evidence of it being a surrogate. And, a p-value of 0.09 in the context of all the missing data without really diving in concerned me. So that's why I voted no.
Longer term, it's harder to predict the
Okay.
exact usage scale.
And I guess I'm the last one to go.
No, I still haven't.
Oh, I'm sorry.
I must be a blind spot too.
It's okay. We're not really ignoring you. We just
more to go.
forgot you were there. Go ahead.
I'll be brief. It's the post hoc analysis. Well, first, I just want to say briefly that I've taken care of a lot of Duchenne patients over the course of my career, from before birth with prenatal diagnosis all the way through to death and participating in autopsies. And I know I've experienced a lot of the pain that the patients and particularly the families are going through. And I'd like to minimize that. And I worry that we might be misleading them by giving approval or recommending approval for a treatment that's unproven. And that's why I voted no.
Long-term strategic flexibility and supply chain leverage.
Dr. Pavlakis.
I agree. I think there are two issues here. One is, and I'm not a statistician, I'm just a clinician that's done much of the trials and been in a lot of NIH committees, but the statistics didn't work. And it was very fragile. I mean, it's possible there is something there, but it was hard to tell from what we have. And also, I think importantly, we don't have a clinical marker that we know means anything, especially on the cardiac thing. And so I think, it's not really clinical and it's not really great statistically. So it's hard to say, yes, let's go ahead with this. And that's why I voted no.
And provide us multiple pathways.
Dr. Vidovich.
Yeah, I just wanted to say it's a difficult decision for me, especially given the profound unmet clinical need. I think the testimonials were actually very, very strong, and that's why it was hard decision to make. But I think after reviewing the data and discussion, I'm not persuaded the evidence is simply there. It's not robust. What I would say is I don't think based on this narrow question, I think we can conclude that the drug is ineffective. It may well be effective in a different trial with different endpoints. And I'm not concluding that the scientific rationale is incorrect. I think that's I don't think you can say about this, but I think there is just not sufficient evidence for effectiveness, and it's unfortunate that for again such a huge clinical need we are unable to provide something. But again, at this time with this data, I had to vote no for this.
Dr. Stuve.
Yes, well, Stuve here. I voted no simply because I found the results presented today insufficiently convincing, like many of my colleagues here. And I want to say it's unfortunate, quite frankly, right now I'm very sad, that I couldn't find the evidence to vote. Yes, I'm a neurologist. During my training, I saw many patients with Duchenne muscular dystrophy. So I also realized that there's a huge unmet need, and it would have been good to see sufficient evidence to say yes, but I didn't see it.
Am I missing anybody online or in the room? I'm good now. Okay. So I'm the last one. This was, I was horribly conflicted on this, and I probably would have abstained, except Dr. Doss said that only cowards abstain. So as a scientist, I would have voted no. As a clinician, I voted a highly qualified yes, because I don't want methodologic issues to close the door on something that could have something there. And I'll tell you what I mean by a qualified yes. Also, as a clinician, I have learned to take seriously the experience of those with lived experience, and also clinicians who day by day take care of patients and just have an instinct that sometimes is just inadequately captured in the studies we designed. And studying rare diseases, I feel, is really, really tough. The numbers are small. They don't follow, as in cardiac symptoms here, they don't follow the typical natural history and presentations that we're used to seeing. It's even hard to have well-controlled patients, let alone numbers, because there's just so many different meds and things going on. So it's really hard. I guess by a qualified yes, I mean, what I'd like is the research to continue, and maybe, and I'm just throwing this out there, this is exactly where you might do a phase 4, meaning there's post-marketing assessment of the patients, and it's very well designed and without any of the flaws that may have been identified, regardless of where the source of the flaws, but really careful post-marketing assessment if it ever went that far. And then based on those data, proper metrics, either continue with giving this as a therapy or withdraw it. But so mine's a qualified yes, though it's probably an abstention. But I'd like to see the work continue. I'd like to see more work on mechanism of action and particularly cardiac readouts that really would be informative as surrogates, given that the clinical picture can be messy. I guess the next question would be for me to pose to you guys whether we on the panel have helped you make any decisions, answered your questions, provided any, can we provide additional insight?
Thank you, Dr. Snyder. I don't think we have any additional questions. Thank you.
Great. Anybody on the panel want to make any final comments or suggestions?
We think that there's a big opportunity, obviously, to self-compute as well. So
Oh, sorry. Yes. I'm going to turn it over to you to make some closing comments. Thanks.
Thank you, everyone. Good afternoon. My name is Megha Kaushal. I'm the Acting Deputy Director for the Office of Therapeutic Products. I'd like to close by offering our sincere thanks to everyone who made today's meeting possible and so productive. First and foremost, a very special thank you that goes to our patients, families, and caregivers who took part in the open public hearing and who shared your testimonies. We hear your lived experiences. We acknowledge the daily realities of living with Duchenne muscular dystrophy, and your voices are indispensable to how we think about the benefit-risk calculus for any therapy in this space. To the patients who enrolled in the clinical trials being discussed today and their families, thank you. The field would not move forward without you. I want to thank the acting AC chair, Dr. Snyder, and the advisory committee members for their time, exceptionally thoughtful questions, deliberations, and recommendations. This is exactly the kind of rigorous, transparent public discussion that helps us do our job well. I also want to thank the representatives from Capricor for the effort that went into their presentation, the FDA review team, including staff from the Office of Therapeutic Products and the Office of Biostatistics and Pharmacovigilance, who worked diligently to prepare the scientific presentation, and the FDA Advisory Committee staff who managed the logistics of bringing this meeting together. Thank you again to everyone in this room and to those who participated virtually. We are deeply grateful for your contributions today, and we value and will carefully consider all of your input. Thank you so much.
Questions from investors about how to think about the mix of debt